Just say something to the teeth.
How much was this projector?
5 something. You see how small it is?
Yeah, but it's strong.
Yeah. It's really small, right?
That's so much.
Yeah,
man. 5? That's how much the regular sizes cost.
Yeah.
But you could take that anywhere. Yeah, once this air, my brothers will be jumping on a plane. Just watch. See this joint. Oh man, you gotta get down.
Go to my page.
What's this?
Might be what is it?
It's an African joint. It's got sustainability.
Oh yeah.
Oh, this is Dr. Ando.
Mm-hmm.
I know Ando. That's the Afro back.
Right. What she mean with her fist?
Come on, take it.
Okay. You should be on. You can share it right now.
Again, it's 2016.
We put this in a few groups real quick. Get started.
Alright, here we go. Share. So message to.
Right. Are you ready? So you know where you ready.
Alright. I'm just sharing it to a couple people. Okay. So, got it on there. Alright. Share now. Send this message. Send it. Alright. Yep. Alright, I'm good to go. Make sure I turn my phone on. I mean, volume down. Cool. I'm ready. Just about to bring it to the beginning.
Ready?
Yep. Good to go. We on?
Yep. You ready?
Yeah.
Hit play.
Yes, sir.
Oh, you want to give us some intro?
Oh yeah, we could do that. Bring it to the very beginning. Yeah, I'm on, right?
Yeah. Hold on real quick.
All right, ladies and gentlemen, thank you for tuning in to Black News 44 Studios. This is our first session, and we have none other, now present them to you, ladies and gentlemen, KT, the Arch Degree. Take it away, my brother.
What's going on, people? I want to thank everybody for tuning in tonight to go along on this journey with me. I want to thank Brother Divine Prospect, you know, of course, Netta, House of Consciousness family, Red and Blue Pill, Mama Pill, everybody who is responsible for this moment here. Tonight, I'm going to be taking everybody who's out there on the frequency. I'm going to take you on a journey into the body, into energy, into this whole discussion. We talk about health, and I'm going to do it straight from the top because I want all of y'all to understand that this is what I do. All right. A lot of people want to know what book I get it from and if it's pseudo or not. Well, guess what? We're going to find out tonight. So just sit down with your pen and pads or your recorder, whatever, and we're going to go on a little journey so we can understand about detox, diabetes, and digestion, dark reactions of the Emerald Tablet. And with that, we can get started. All right, so we are all electrical beings. Now, what does that mean? That means when we go deep inside ourselves, we find out that every process that our body goes through is electrical. So I like to present a Greenleaf Herbal Solution presentation in association with KTL Media Oceans 14. This represents your last drug, the last sale. Instead, it's time to be the cell. All right. Now, as we go inside the cell, we need to understand what a cell is composed of. It's composed of a cellular membrane, which represents the brain and the skin of the cell, the connection with the external environment. We also have ourselves a cytoplasm, which represents the blood inside of the cell, and the DNA inside of the nucleus, where all the genetic information is at. I mean, this is stuff we all know, right? You got prokaryotic and eukaryotic cells, okay? So we're going to focus on the eukaryotic right now. The eukaryotic cell has organelles and a nucleus. All right, this is what makes it different from the prokaryotic. Eukaryotic cells are plant cells as well as animal cells, and of course, man. All right, the prokaryotic cell doesn't have a nucleus or organelles. Its genetic structure is free inside of its cytoplasm. All right, it has proteins to do the work, but its genetic material is free. All right. And they also reproduce through asexual reproduction. All right. Bacteria is an example. So as we go back to eukaryotics, what are organelles? Organelles are little organs. So a cell represents a microcosm of you. Organelles represent the organs and their systems in the body. So we're going to start going through some of the organelles. First thing we're going to look at as we go inside of our cell. Deep inside, we're going to go on the nucleus and we're going to find DNA. Every cell has 6 feet of DNA, meaning that all the DNA combined can go back and forth to the sun 300 times. That's how much DNA we got in our body. All right. And that's a library. OK, it's all enveloped in the nuclear membrane. And deep inside, we have the wound up versions of the chromatin, which is called chromosomes, where the genes are. All right. Also, you have a bundle of DNA, which is called the nucleolus, and this is where ribosomes are produced. Ribosomes are the organelles responsible for synthesizing proteins. All right, so right now you see ribosomes exiting the nuclear pore of the nuclear membrane of the nucleus, and we are right now inside of the cytoplasm, a gelatinous fluid substance that is the ether, or the space in which everything takes place. It's the watery environment. Okay. Now, from the cytoplasm, where we're going to go next is another organelle called the endoplasmic reticulum, which is broken into 2 types. We have the rough endoplasmic reticulum and the smooth endoplasmic. The rough is called rough because it's studded with ribosomes, because this is the site that proteins are made. Smooth is where the fats are made. So right here, you're seeing inside the rough how the ribosomes have generated peptides, and these peptides are getting formulated into proteins. All right. As they get formulated into protein, they are secreted or excreted through vesicles. These vesicles then go from the rough endoplasmic reticulum to the Golgi apparatus. The Golgi apparatus finalizes the packaging like FedEx to let these particular proteins know where they're going to go in the body. They also add other pieces to it like lipids and sugars in order to upgrade the particular protein into other forms. And in this case, you see that it changed the structure to make it more tightly bound, and it added a lipid molecule, which is another name for fat, to it. All right, this is a carbohydrate molecule that got connected to that one. So when you deal with eukaryotic cells, you got animal cells and plant cells. All right, plant cells have a central vacuole. That's this big reservoir of water because plants need water. We're going to explain why shortly. All right, we have lysosomes. Lysosomes is the digestive center of the cell. This is where digestion takes place. All right, where you break down toxic chemicals or particular things that you need in your cell. They break it down into smaller constituents. Mitochondria is the powerhouse of the cell. This is where the energy is made through the electron transport chain, more specifically ATP, adenosine triphosphate. This is made through ATP synthase, through an electric gradient. It's called cellular respiration, and this is how we get the energy in our body. They have their own DNA and they multiply. They multiply throughout our body when we need them, so we have more energy. Okay. Also, as we go further, we have a cytoskeleton made up of microfilaments and microtubules. This represents not only the skeleton of the cell, but also the nervous system, because the microtubules are made up of alpha and beta tubulin, which represents binary code. Okay, now we're going to go into the plant. The plant is amazing. This is where we're going to spend first of our real time going deep into the science. We go inside the plant cell. The plant cell has a cellular wall as opposed to the cellular membrane. And inside inside of the cellular membrane, I'm going to pause it right now. Let's get a pause. Pause. Yeah. Okay, so this is going to be our first break. So we're going to kind of stop and go for a little bit right now. Now, when you go inside the plant cell, you find inside of the plant cell these organelles that are called chloroplasts. The chloroplast is actually its own cell. All right, it's its own cell because it has its own DNA. All right, and what happens in the chloroplast is photosynthesis, and this is where we're going to really spend the bulk of our discussion tonight, because the conversation in regards to health is really a stupid argument that we constantly have in regards to what we should eat, whether we should be eating blood, flesh, and starch, or whether we should be eating live electric foods, which is a conversation that was introduced to us mainly through Dr. Sebi years ago. We're talking 40 years ago. Perpetuated through my mother, and in the community we're still talking about it now, but there's a lot of confusion going on because people think that it's not beneficial to focus on the plant kingdom. Well, I'm going to show you tonight what electric foods actually means. All right, so you know why it's important for you to get your greens in. It's not just conversation, talk, or words anymore. It's about the science and the genetic consistency, because that's another thing Dr. Sebi introduced to our vocabulary in our conversation in the 80s was food gene consistency. Okay, Asians, Russians, Italians, they all came here with their food. Did we? We took the scraps when we were prisoners of war during the slave trade, but guess what? We ain't in that condition no more. We can grow our own foods in our backyard. We have Google and the internet to study all types of information. There's no excuses for us to be eating chitlins. I'm sorry. If you want to eat it, but don't knock me because I'm coming with the right information to help my people, help you, help myself, help my family. So we go inside of the plant cell, and inside of the plant cell, we have the chloroplast. And when we go inside the chloroplast, we have these structures which are called thylakoids. Okay, thylakoids are these disc-like structures. All right, when they're stacked on top of each other, they're called grana. They're connected by bridges called lamellae, all right? And these lamellae increase the surface area and also connect one thylakoid and one grainer to another thylakoid and another grainer so that it can utilize the process of photosynthesis more efficiently, all right? Now, the thylakoid are membranous sacs, all right? And these membranous sacs are the sites that photosynthesis is going to occur. You can let it go a little bit. All right, so let's go inside. So thylakoid, okay, we have the lumen, which is inside, and the membrane. All right, we're gonna go inside a little deeper, okay, just a little bit deeper, so y'all can understand what we're dealing with. This is the membrane, okay, and inside the membrane you have a structure called photosystem 2. Okay, pause it real quick. Yeah. All right, so if you see with this membrane, you have a layer of a membrane here and a membrane here. That is called a phospholipid bilayer. Phospholipid bilayer is exactly what it means. It's made up of phosphorus, it's made up of lipid, and it's bi, which means 2 layers. The reason why it has phosphorus and lipid connected is because you want to have a hydrophilic chemical and you want to have a hydrophobic chemical. Because when you have hydrophilic, that means water loving. That means it's a side of this particular construct that will deal with water and water only. All right. Then you're going to have hydrophobic, which means water fearing, meaning that it's going to point away from water like oil. So the hydrophobic will be here in the center in between the 2 layers, and the hydrophilic will be pointing into the aqueous solutions, which divides external environments from internal environments like a door or a wall. All right. Studied inside of these membranes is photosystem 2. Or rather, I'm not even going to say photosystem 2 yet. I'm just going to say that you're going to find different proteins that are going to be inside of the membrane that regulates what comes in and out of the cell. So anything and everything can't come in, because if you didn't have a door in your house, if your windows did not, when they were not shut, then anything and everything can come in and out. And you end up having the door and you have the window so you can prevent anything from coming in and out, meaning that your home is selectively permeable. You're only going to allow certain people to come in there, certain energies to come in there, and you're responsible for letting things go. And that's what this membrane is for. Now, photosystem 2 is the first site that we deal with in photosynthesis. Now, what is photosynthesis? What is so important about this thing? You know, I'm going to let you know that photosynthesis is the most important study in technology right now, because the information that I'm going to give you tonight, when you go through the research, is so scarce. It's so scarce, however you want to enunciate it. As you dig deep till 4 in the morning, that's going to be around the time when you start really finding stuff. Because this is what they're studying in the most advanced colleges, because this is the answer to the energy, quote unquote, crisis that we have. And you're going to see why it's so important. This is why I wanted to talk about this, because I come from Greenleaf Herbal Solutions. You got problems, we've got solutions. That's what we deal with. Now, photosynthesis deals with 2 main things. The first thing that photosynthesis deals with is the production of oxygen. All right, molecular oxygen. Why is that so important? I don't know. Maybe the fact that you can go months without eating. You can go days without drinking. How long can you go without breathing? Where do we get our air from? In the atmosphere. But what in the atmosphere, specifically oxygen, is what we need out of the atmosphere in order for all of us to breathe. And we get this oxygen from the plant. So that's the first thing that it does. And we're going to talk about how it does this. The second thing that photosynthesis is responsible for, which is why I named it Dark Reactions of the Emerald Tablet, because the emerald tablets are the thylakoid, is the dark reaction. It's actually called the dark reaction. And the dark reaction is the process where an enzyme takes CO2 or carbon dioxide, our waste, Now, when I say waste, I want you to understand what waste mean. I mean waste like you just got done throwing a party. Everybody just took the trash outside. You got about 8 bags of trash out there. When I'm talking about waste, I'm talking about you sitting on the toilet after you just done had some Indian food. You done blew it up. I'm talking about waste where you just drunk a gallon of water and you headed to the bathroom. Whether you're sweating, whether you passing gas, burping, vomiting, whatever, that is waste. Plants take our waste, carbon dioxide, in a gaseous form and change it from a gaseous state to a solid state, a state called sugar that we use for fuel. Now, there is not a more sustainable process that exists within our universe other than this photosynthesis. And we're going to go through the light reactions of oxygen production and the dark reaction of what they call carbon fixation. So let's go on. All right, so this is photosystem 2, okay? Photosystem 2 has chlorophyll molecules embedded in it so that chlorophyll A and chlorophyll B, so as sunlight comes down, right, it excites the chlorophyll molecule. Right in the center, you have something called the reaction center. Pause. Remember what a photon is, okay? Photons are particles of light that come from the sun. Remember, the sun is 93 million miles away, okay? Remember I said 6 feet of DNA, we could go to the sun and back 300 times, right? 93 million miles, light comes from the sun, and it beams our planet. And when it beams our planet, we see color, but color only represents a certain fraction of light. If you look at the electromagnetic spectrum, it's broken up into nanometers. Nanometers means the millions of a meter, and that's real, real small. The smaller the nanometer, the higher the frequency, because that means there's more energy that can be compacted inside. The larger the number, the slower the frequency, the less information in there. So when you think of color, the visible spectrum, you're going from red to orange to yellow to blue to indigo to violet. The red is going to have the largest number, all right, of 700. And as it goes down the line to 400, 300, it's going to go into the purple, all right, or the ultraviolet energies, all right. Now, when the light comes and we see a color, for instance, we all see my red shirt on the screen or my yellow hat. The reason why my shirt is red, the reason why my hat is yellow is because my shirt is absorbing all colors of this spectrum except red. And it's creating a mirror where it's reflecting the color red back to you. So you are recognizing the reflection, all colors but what is being absorbed. The same thing with my hat. My hat is yellow. You're seeing every color being absorbed into this hat, but yellow reflecting back to you. So when we see plants are green. That means that they're absorbing the red and blue spectrum of color and reflecting green back. More specifically, there's frequencies of 680 and 700 nanometers. So 93 million miles away, the sunlight comes beaming down into the plant, through the plant cell wall, into the chloroplast, through the thylakoid membrane to hit this photosystem to charge the chlorophyll A and chlorophyll B molecules so that the electron inside gets excited. When electrons get excited, they jump their orbital. Now, what do I mean by orbital? What is that about? Well, we all know atoms, and atoms' nucleus consists of protons and neutrons glued together by gluons within inside the nucleus of the atom. And around it, in its peripheral, in its extremity, is the electron. Well, if I have an oxygen atom, which consists of 8 electrons, well, the first valence, so the first orbital can only hold 2, while the second can hold 8. But it's 8 in all. So if there's 2 in the first and there's 6 in the second, there's none in the third. So when I say that a molecule gets so energized, or an atom gets so energized that the electron gets excited and jumps orbitals, that means it leaves from the particular orbital it is and goes to the next one, which makes it kind of like free. It's up for grabs. An electron is really an embodiment of God because it represents the power, the electromotive force that exists in our universe. Everything that we deal with is based in electricity and magnetism. So the electron represents that force that is necessary. All right, so let's keep going. All right. So once again, the photons come in, they start to charge this photosystem 2. As they charge this photosystem 2, many reactions are going to occur, and we're going to talk about all of the reactions that take place. Remember, we're talking about the light-dependent reactions right now, all right? Now, the most important mechanism or process that takes place is the splitting of water, which is an amazing process because it takes so much energy to do it in a lab. And in splitting water, we create, as I said before, oxygen. All right? This is what happens in photosystem 2. And we're going to talk about how this happens. So as you see the photons raining down from the top, you're seeing the splitting of water at the bottom and the protons. All right? So this is, we're going to go through a couple slides now because we're going to go a little bit more deeper into the process before we go back to the animations. This is the, let's go back real quick. We're gonna pause on each slide. We're gonna pause on each slide. Bear with me, people. There we go. All right, so once again, thylakoids are these discs shaped like coins. These are the emerald tablets, all right? These are stacked on top of each other and they are called grana, all right? Now, if you think of mitochondria, mitochondria actually means grain. It means mighty grain. And this is interesting that in this particular organelle called the chloroplast, this is called grana. It's nothing more than the inner membrane of the chloroplast folded in on itself, forming these discs, all right? And then you have the lamellae, which are these bridges. And how I could describe it is, let's say we in the projects, right? And you got, I don't know, maybe about 6 buildings in the projects. You know, in New York, they up. In Atlanta, they kind of spread out. But in New York, they go straight up bricks, right? And let's say that you have one brick building. Let's say we got brick building A, and brick building A on the 5th floor want to connect to brick building C on the 7th floor, okay? Or you got brick building D connected to brick building F from the 6th floor to the 8th floor. All right, that's what these lamellae do. They connect each one of the grana to particular thylakoids in each one, like I said, to increase the surface area and so the energy can get propagated more efficiently. All right, the photosystem and the protein complexes we're going to talk about are going to be embedded inside the membrane. All right, the membrane. Okay, so the stroma represents the area outside of the grana and the thylakoid, which is in the chloroplast, but outside of these. And then the lumen is going to be the space inside of each one of these particular discs here, with the membrane being the membrane that actually envelops everything inside. Let's go to the next slide. All right, so I'm going to give everyone a brief rundown of the process, and we're going to keep going through each one. Let's pause right here. All right, so this is the membrane of the thylakoid, okay? This is that emerald tablet. And when they zoom in on that, you see that it is a phospholipid bilayer, okay? This is the phosphorus side connected with some chlorine, and it ends up creating this head. And then the tail on the bottom is the lipid. This is the fat. So this is the hydrophobic, this is the hydrophilic. Because if you see, this is the lumen. And the lumen has all the aqueous water material inside. So the water is what's going to touch that side. And then this is the stroma inside of the chloroplast, the organelle that we're talking about. And that's all water. So it's facing that way. But the lipids are aggregated inside to keep it divided, okay? So as the sunlight comes down, it's gonna hit photosystem 2. Photosystem 2 is surrounded by the LHC, which is called the light harvesting chlorophylls, which are surrounding the reaction center. All right, the light comes down and it hits this reaction center, which is called P680. It's called P680 because 680 represents the nanometers. It represents the frequency of light that's coming in specifically. All right, so as it comes down, it's going to end up exciting the chlorophyll. As it excites the chlorophyll, the electron gets excited, it jumps out. When it jumps out, it starts going through a chain. And the name of this chain, you could call it the electron transport chain, but on a bigger scheme of things, it's actually called the Z scheme. Now, the first thing that the electron is going to do is it's going to jump down to a complex called pheophytin, and from pheophytin, it's going to go through plastoquinone. From plastoquinone, it's going to end up going to the cytochrome complex. From the cytochrome complex, it's going to go to plastocyanin. From plastocyanin, it's going to go to photosystem 1. When it goes to photosystem 1, this electron that's been transported through this whole system has lost a great deal of energy, so another photon of light is going to come in and re-energize it. When it re-energizes it, guess what? It's going to go back up. It's going to hit ferredoxin. After it hits ferredoxin, it's going to hit NADP reductase. And then from there, it's going to create a molecule called NADPH. And then it's going to go on to the dark reactions. At the same time that's going on, as this electron is flowing through the system, it is attracted to hydrogen protons, because remember, an atom consists of protons and electrons. Protons and electrons is what makes up an atom. The same way a north part of a magnet and a south part of a magnet come together and they're attracted, so is electrons with the nucleus or with the proton. So as this electron, as it got split and is getting carried by these carrier molecules through this membrane, guess what? The hydrogen that it got split from is going to get pumped. It's going to get pumped into the area of the thylakoid, and the hydrogen ions are going to get abundant. It's going to go from an area of low concentration. The high concentration is going to build up. And as this hydrogen concentration builds up, because this membrane is selectively permeable, like I said, it can't get out into this area where the low concentration is at. So it stays built up. So whenever you have a high amount of electrons or negative energy in one area, and then you have a high amount of positive energy in another area, you got a battery. It's called an electric gradient. So this is dying to get out here, but it can't go through here. So that's where this mechanism, ATP synthase, comes into play, because ATP synthase provides a channel for the hydrogen ions to go through. They dying to go through it. And when they go through it with such a magnanimous force that it makes it spin at light speed so fast that it fuels this motor into doing its job. And what its job is able to do is to take a molecule of ADP, add it to inorganic phosphate, and make ATP, which is the energy batteries of your body. So this is how the cell is, the cell is able to produce the energy it needs to sustain as a byproduct of the electron flowing through this chain. It turns it into a hydrogen pump. All right. So that's just a quick overview. We're going to go through each one. Let's go to the next slide. All right. All right, pause. So this looks like a bunch of macaroni noodles, probably, to everybody out there checking it out. But this is actually a composite of photosystem 2. I'm going to give y'all different pictures because I want y'all to see the technology that is sitting right outside your door. You know, as a people, we are sleeping on one of the most efficient forms of fuel and energy that we can use, but we can't use it until people understand it. So I don't know who's watching, but somebody's going to watch and it's going to go off in their mind, and then they're going to be part of this movement as well. That's why I have to share this information. So, as I said, this is photosystem 2. What you see right here, this orange and this blue and these greens that are coiled up, they represent the pigments, the pigment centers. And the pigment centers are not just chlorophyll with green. They actually consist of beta carotene and other things because they represent other nanometers or other frequencies of light. So it's not just about the 680 and the 700. It also deals with other frequencies in between. And you see the beta carotene and you see the other colors in the autumn. You see what I'm saying? When the magnetic field changes on our planet or where we are in respect to the sun, and the leaves start turning colors, we start seeing burgundy. You know, we start seeing orange and yellow, and those are the other pigments that start to reflect. Let's go to the next slide. All right. Photosystem 2. Okay, pause it. So yeah, so photosystem 2, we have the sunlight. These are the different pigments. What we just saw in the last slide with the different color coils. Okay, you got chlorophyll B, you got lutein, you got zeaxanthin, you got beta carotene, we got lycopene, which is like found in tomatoes, you know, chlorophyll B, chlorophyll A. All right. And if you notice, you see P680, P650, P510, P500, P480, P470, P450. All of those represents frequencies of energy. That's electricity. They're all named by their electrical identity and their electrical signature. And the reason why the arrow's going down is because as it goes through the system, the electron is losing energy as it goes because it's losing heat. See, in each energy transfer, some energy is lost as heat, which is the second law of thermodynamics. But enough energy is passed to P680 to eject an electron to the electron transport system. All right, so 450 is very high frequency. Remember I said the lower the number, the higher the energy. So it starts off real high, but as it declines in energy, it declines just in time for it to stimulate chlorophyll so that an electron can jump. Now, as this energy is getting utilized, these electrons are getting used, and it's like electrons are not just going to keep popping out of nowhere. So where are they coming from? Where they're coming from is from the water. What if I told you it's not plants that give us oxygen? Plants are actually our gills. Man, what is KT, the arch degree, talking about? Let's go to this next slide. Plants are our gills, meaning that a fish uses gills for what? They swimming through the water, and in order for them to breathe, let's pause it right there, the gills are able to separate the medium that they in so that they can breathe and they can use the oxygen that's in the water. Well, we breathe water too. Plants are just responsible for splitting water and ejecting the oxygen molecule in a gaseous form out. But it's really water that provides, or rather is the source of the oxygen that we breathe every day. It's the water, not the plants. The plants are the technology we use, or the gills we use to breathe. It's the water where we get the breath from. All right. So this once again is photosystem 2. This is a little bit a more detailed description of it. And these right here represent the different chlorophylls. Okay, the greens. All right. The orange represents the beta carotene. All right. And you're going to have quinone, which represents a transport molecule. We're going to go through this visually so you can understand what all of this is. This right here is called a manganese stabilizing protein, and this right here is called the water oxidation site. This is out of photosynthesis. This is the most discussed technology on the planet right here. They got all us looking at CERN. Forget CERN. CERN ain't got nothing on the water oxidation site. Or they also call it the oxygen evolving complex. This is what ties, this is how we get a better understanding of evolution. And as I get through this, I'm going to give you all a better idea on what I was talking about at the evolution conference as well. Now, here you got something called, as I said, the oxidation evolving complex, or the water oxidation complex, which consists of manganese, calcium, and oxygen, and they create a structure that is responsible for splitting water. All right, let's go to the next site. What is so important about splitting water? Splitting water, if we were to do it in a lab, takes crazy amounts of energy. so much amount of energy that the amount of energy it would take to split the water would not equate to the energy we get out of splitting it, which is kind of like everybody's eating right now. When y'all go invest in a Big Mac, that's like withdrawing $3,000 from a bank that got 15 cents in it, because the amount of energy invested in trying to digest, which is not going to happen, but you're going to try to digest that Big Mac or chicken nugget or whatever that chicken garbage that you're trying to eat is, you're investing too much energy in the digestive process in respect to the energy that you're going to get out of it. That's like you getting up, going to work, working a 40-hour work week, and when it's time to get your check, your boss is looking at you like, what you want? You ain't getting nothing from me. That's what you're doing to your body every day. All right, so once again, photosystem 2 complex. All right, and as we zoom in this area right here, this is the water oxidation area right here. So I'm gonna read this excerpt right here. So the arrows within the PS2 indicate the direction of electron transfer, which comprises the following redox active cofactors. What does redox mean? Okay, when you're dealing with redox, you're dealing with reduction and oxidation. Okay, reduction deals with the gaining of an electron. Oxidation deals with the losing of electron. And how you remember that? Just think of the phrase oil rig. Oil rig. O-I-L-R-I-G. Oil is loss. Reduction is gain. That's how you remember that. Okay, so redox active cofactors: inorganic, manganese 4, calcium, oxygen 5, right? That's what this structure is right here. Cluster, redox active tyrosine. Where do we hear the word tyrosine? Every time we talk about melanin, because tyrosine is the prerequisite for melanin. Tyrosine is also the preliminary to T3 and T4, which are the main hormones for the thyroid gland, which represents anabolic and catabolic reactions inside of the body. All right. And once again, here it is. It's the mechanism that is responsible for reducing the properties inside of photosystem 2 to get started the process of splitting water. All right. The primary electron donor, P680, that includes a pair of chlorophyll A, okay, which are labeled PD1 and PD2, right up here. If y'all look, y'all find it, and 2 accessory chlorophylls. All right. The primary pheophytin is the first electron receiver or acceptor, meaning as the photon comes down and excites the chlorophyll gets excited, dumps an electron. The first person to receive that electron, the wide receiver that catches that pigskin as he's running is the pheophytin. Pheophytin is the wide receiver. He catches the electron. We got to think of the electron as a football. It's a hot potato in the game. All right. Now, the primary QA, all right, and the secondary QB, quinone acceptors. So after pheophytin catches it, a bunch of cats is about to get him, so he got to throw the ball to the side to QA and QB. QA catches it first, right? And then there's a cat in between QA and QB, which is represented by iron. Okay, so iron is in this equation too. QB catches the ball. We're still trying to make the touchdown. The touchdown is the splitting of the water molecule. All right. Quinone acceptors, the phytotails of the chlorophyll and the pheos and the isoprenyl chains of the quinones have been cut for clarity. They're just saying that they cut those things out so you can see what's going on better. The light-induced S state transitions of this complex cluster are indicated by arrows with HV labels. Okay. So what they're saying is as the oxidation of water takes place. Oxidation of water is the splitting of water. Remember I said oxidation is the loss of electron. So if you have H2O and it loses an electron, H2O can't get together anymore. It has to split up. So hydrogen goes this way, oxygen goes that way. And the process is this cycle right here called the KOK cycle. And it is represented by these different S's right here, S1, S2, S3, S4, S5. Each one of these stages represents a particular reaction taking place in regards to the water splitting. All right. So the PS2 structure and the Zoom structural model of the manganese calcium oxygen cluster in the center of the KOK or Koch cycle are based on the recent PS2 crystal structure at a resolution of 1.9 angstrom. So the funny thing is they call it the Koch cycle. And the deeper we get into microbiology and really study our sciences, we start to find out how many Kemetic names are in there. Because Kook and Kuket, that's part of the Ogdo. It's going to always come back to the Kemenu over and over again. All right. So once again, the light comes down and it hits this reaction center. Okay, really it bounces around amongst these chlorophylls first before it hits the reaction center, excites the electron to pop off. It gets received by the pheophytin onto the quinones, on to the next structure, the next structure, the next structure along these chains. But the first process that we have to pay attention to is the splitting of the water, because splitting water creates oxygen molecules, and oxygen molecules is what we breathe. Let's go to the next slide. All right. Pushing on, pushing on, pushing on. And let me know how my time's looking too. All right. All right. So I'm going to read this too. In nature, the splitting of water to molecular oxygen is catalyzed by the membrane-bound pigment protein photosystem 2 of plants, algae, and cyanobacteria. The catalytic site of the water-splitting reaction is an inorganic tetramanganese monocalcium pentaoxygen. Oh yeah, that sounds like a light. Tetra, we all know what that is, right? Tetragrammaton, tetra, whatever. Tetra means 4. All right. So tetra means 4. So 4 manganese. Okay, 4 manganese. Monocalcium. What is mono? What's a monologue? What is monotone? That means 1. All right. So that means 1 calcium. Pentaoxygen. What is the pentagon? What is a pentagram? Huh? That means 5. Okay, that's why here you see Mn4Ca and O5. So we're talking about 4 manganese, 1 calcium, 5 oxygen creates this complex cluster that forms together with its protein ligands the water oxidizing complex of PS2. All right, water splitting by this cluster is energetically driven by the strongest biological oxidant, 680, P680. All right, with the midpoint, we don't got to worry about that. Generated by the light-induced charge separation within the chlorophyll-containing reaction center of PS2. Okay, a redox active tyrosine residue, there go the tyrosine, is known as YZ, is the essential electron transfer intermediate between the photoactive reaction center and this cluster of PS2. Following light absorption, the cluster is oxidized stepwise, 1 electron at a time, and thereby cycles through 5 redox states known as SI states, which I showed you, the Koch cycle, right? Where I reflects the number of oxidizing equivalents stored by the cluster. That's what the I stands for. The 4 electron, 4 proton oxidation chemistry of 2 water molecules is completed when the 4 oxidizing equivalents are accumulated within the water oxidation complex. And the highly reactive S4 state relaxes into the most reduced S0 state with the concomitant 0-0 bond formation and release of O2. Okay, so that sounds like a lot. To sum that up, they're just giving you the step-by-step process on how light comes in to chlorophyll, and through photosystem 2 and this reaction center, they split the water so that we could breathe every day. You understand what I'm saying? Next slide. This is why he said electric foods. You're not going to find that in a steak. All right, so this is the water complex right here. All right, this is one composite of it. Let's go to the next one, because the next one I got labeled. All right, and we'll pause on the next one. It kind of looks like a chair. Yeah, here we go. So here's manganese. Okay, it says W1, W2, W3, W4 because these are where the 4 hydrogen molecules at. You got to have 4 hydrogen molecules in order to create 2 O2s because H2O. You know, there's 2 hydrogen in there, but there's 1 oxygen in there. But we need all these water so we can create the 2 oxygen molecules in there. We got manganese 3, manganese 2, manganese 1. O1, O3, that stands for oxygen. Ca stands for calcium. So this is the technology. Okay, there's two powerful technologies that are in photosynthesis. This is one of them. I want y'all to soak that in. And they call it a chair, but it's a throne. So we might be dealing with our set right now. And then when I get with the other complex, we're going to be getting into a saw, who a saw is. But this right here represents a throne. And this throne is what is responsible for splitting water so we can breathe. All right, let's go on to the next one. All right. Okay, now let's pause. I'm going to show two versions of this. This is the Z scheme. I'm going to keep reiterating this information because it's kind of upstairs. I know y'all don't hear about photosynthesis every day, but I want y'all to get it. All right. So energy of an electron, we deal with lower and higher. If the arrow's going down, that means it's a lower energy coming out. If it's going up, that means it's excited. All right. So we're going to start over here with the photosystem 2 complex. This is P680. 4 photons come from the sun. They hit the photosystem 2. By doing that, it uses up its electrons, but it's going to replenish those electrons through the splitting of water. 2 H2O molecules end up yielding 4 hydrogen and 1 oxygen molecule. So we can breathe. This 4H plus ends up creating that hydrogen, increased hydrogen gradient I talked about earlier that will fuel the ATP synthase to make ATP. Now, those electrons go up to the first acceptor, the wide receiver, catching the football, field fighting. Field fighting passes it off the plastoquinone. Plastoquinone passes it off the cytochrome complex. As this is occurring, ATP produced via proton motive force, okay? Because of the hydrogen building up, it's causing the ATP synthase to create ATP. From cytochrome complex, then it's passed on to plasticinin. Plasticinin is the midway point in photosynthesis. From there, it goes to photosystem 1, which is P700. Okay, it's a little bit lower, but it has to be lower because look at the direction of the energy. It's starting to decline. This is a larger number, so it takes less energy. More photons comes in, excites. The electron gets excited again. Now that it gets excited, it goes through another electron transport chain where it goes to ferredoxin and it goes to FNR, and in there it ends up creating NADP. I will explain who NADP is and why he's so important very soon. But they call this the Z scheme because it goes like a Z. All right? It makes a Z. And if you can visualize the Z, you can visualize the process. Let's go to the next slide. Electric foods, people, chlorophyll, people. We got to get more greens in our diet. You want to keep eating meat? Go ahead and do it. But that don't mean you can't put some plants along with it. All right? Drink some green juice. Get some water in. I'm telling you, you're gonna know the difference. Okay, Z scheme of electron transport and photosynthesis. Okay, once again. All right, so here's that complex right here. Magnesium 4, 1 oxygen, right? And the calcium, right? And then we're dealing with the tyrosine residue. Okay, that is what is responsible for splitting the water. All right? Now, this is the chlorophyll, the 680, excited state because of the photons. Thiophytin is the first acceptor. Plastoquinone A to plastoquinone B. Now, right here, you see HCO3 and CO2 because carbon dioxide is utilized within photosystem 2 to make bicarbonate because it acts as an intermediate between these two quinones right here. And as you see, as this goes on, the hydrogen is going to increase, all right? So it goes on to plastoquinone, cytochrome B, cytochrome F, all right? We got iron and sulfur here as an intermediate between those two. Plasticinin, once again, photosystem 1, which is P700, excited again. Then we go down to this chain right here with the ferredoxin, down to the FNR once again to create the NADP. And right here is just a breakdown of all of that. I'm not going to read it. Y'all will be able to pause it when this thing get uploaded or you get the copy, and y'all could go more in detail into reading it. But I went over it a few times. So let's go to the next slide. Once again, Z scheme. See the Z? Z scheme. All right. All right, pause. Okay, so this is what NADH is all about. All right, so remember, electron acceptor, plastoquinone, and all of them represented um, um, electron acceptors. NADP represent electron carrier, like it can hold it, and it's not just arrested to the membrane. It can move around wherever it want to go. Okay, so it's similar to somebody catching, wide receiver catching a football. You know, he ain't just limited to one play. He could do whatever play he wants, whatever is needed to make the touchdown. This is what the NADH is. So one, NAD plus, which stands for nicotinamide adenine dinucleotide, within a cell, along with two hydrogen atoms that are part of the food that is supplying energy for the body. All right, this is it right here. Here's hydrogen, and it comes over. NAD represents an empty state. No one's in the cab. Okay, these represent the two hydrogens hailing the cab. All right, once they get inside the cab, now NAD is loaded. It became reduced because reduction deals with what? The gaining of electrons. So it gains some electrons because now they're inside of the NADH, so the cab driver can take them. See, proton is oxidized, but NAD gets reduced because the hydrogen lost an electron, but the NAD gained an electron. NAD is reduced to NAD. NAD plus is reduced to NAD by accepting an electron from a hydrogen atom. It also picks up another hydrogen atom to become NADH. From that process, the NAD can then take these electrons and dump them off to go pick up more electrons. So these are used in later stage of respiration. These are used in later stage of respiration. So NADH reduced, nicotinamide adenine dinucleotide, carries the electrons to a later state of respiration, then drops them off, becoming oxidized to its original form, NAD+. Next slide. So that's all NAD is, the cabs, picking up electrons, dropping them off. All right. So that's what the light reactions is responsible for doing. It's responsible, oh yeah, we're about to get into it now. It's responsible for splitting the water and then creating ATP, energy for the cell, and creating NAD, because it's ATP energy and it's NAD are going to be used in the other part of photosynthesis, which is the dark reactions. So half of it is made for us breathing oxygen, the other half is for the dark reactions. Now, when I was at the evolution conference, majority, about 90%, I'm going to say 95% of all the feedback I got back was a lot of people who understood what I was talking about. My most revered and respected commentary was from A.A. Rashid. I really respect my brother. He's my big brother because we go way back, and he, you know, I didn't even get to really build with him yet. It was really by way of Red and Blue in regards to how much he really appreciated what I presented. Now, this is what I was really talking about, because I talked about the acacia tree, and more specifically from the acacia tree, the exudation of the gum that comes out the acacia tree, which is called arabinose. Arabinose is the original name for ribose. I'm going to say that again. Arabinose is the original name for ribose. Arabinose is the sugar of the gum arabic. Okay, gum arabic is called that because arabinose is called that because it was isolated from the acacia tree, from the gum out of the tree, which I call kaminose because it's called Kami and Kemet, which is the same name as the people. Now, ribose is the name that was created off of arabinose. Now, why do they call it gum arabic and arabinose, you know, that whole Arab thing? Well, Kemet was producing it down in the Sudan, down in Upper Kemet, all right? Arabs represented the merchants, the people that would take the product that we would create it and sell it to the European, because we wasn't dealing with him. He would do all that work. So when the European would deal with the merchant, the product that they received, they named it after the merchant. So they would call it gum arabic because that's where they got it from. All right. So then that went into the arabinose, but from arabinose, they ended up creating the term ribose. Now, why is ribose so important? Well, if you're talking about our DNA, which stores all of our information, you're talking about deoxyribonucleic acid, which deals with ribose. Or if you're dealing with energy, you're dealing with adenosine triphosphate, which deal with adenine and ribose being connected. And if you're dealing with the dark reaction, you're dealing with ribulose biphosphate and an enzyme that we call Ribisco. Now, I know this is not a term that has been used in the community yet, but when we get done, you're not going to forget about them. This is the #1 enzyme on the planet, #1 protein on the planet, most plentiful enzyme on the planet. What makes this enzyme so important? Let's keep going through the slides. What makes this enzyme so important is it is single-handedly responsible for taking the gas of the waste of carbon dioxide out of our atmosphere and fixating it into sugar or into a solid form. I'm going to say that again. Carbon dioxide is a gas. We use sugar as a fuel, and it's also a basis for a lot of things that go on in our body, you know? Communication with our cellular membrane. That's how we deal with our environment because of the sugars that are connected or inundated throughout the plasma membrane. And what is responsible for making the sugars in the body? This enzyme Ribisco, because Ribisco is what is able to fixate carbon fixation. Now, let's go to the next slide. Dark reactions. They don't call it dark reactions no more. They call it Calvin cycle because a guy named Melvin Calvin was responsible for creating it. Pause it. And the reason why they say they don't want to call it dark reactions anymore is because they say, oh, it happens in the light. Well, no shit. Yeah, it's photosynthesis. It happens in the light. It all happens in the light. But that ain't why you call it dark reactions. It has nothing to do with it being in the dark. The reason why they call it dark reactions is because it deals with carbon fixation. And if it deals with carbon fixation, we're talking about carbon, the bases of melanin, carbon, the bases of soot, charcoal, coal. It's the blackness, the melanin. So yes, it's the dark reaction. All right. So of course they want to get away with that term because anytime in science we could take anything and equate it to our people, equate it to who we are, equate it to knowing more about ourselves. They always want to switch it around and name it something that would keep them on our mind. But in this case, I know why it's called dark reaction. It's called dark reaction because of the carbon. So we're going to go through the dark reaction. This is the second part of photosynthesis. All right, so carbon dioxide comes out of the atmosphere. When it comes out of the atmosphere, there is a molecule by the name of ribulose biphosphate. Ribulose biphosphate ends up getting fixated to carbon dioxide by that enzyme Ribisco. Once it gets fixated by that enzyme Ribisco, it ends up going into a 6-carbon intermediary molecule that's unstable. It's so unstable that it doesn't stay together. It splits apart. It splits into 2 equal parts of 3 carbons. As you see right here, these 3 carbons are called 3PG, or rather phosphoglycerate. All right. The phosphoglycerate then, as it comes down, this cycle gets hit with ATP. As it gets hit with ATP, it's giving another phosphate because ATP really gives energy by giving away one of its phosphates. Because ATP stands for adenosine triphosphate. Tri means 3, di means 2, M is for mono, which means 1. So if I go from ATP to ADP, that means I gave 1 up. I gave the phosphate up to this group, which you see down here now. You see 2. So now it goes from phosphoglycerate to biphosphoglycerate. Okay, once it goes to once it goes to the biphosphoglycerate, then the NADPH recreated. Remember, the ATP recreated from the light reactions. The NADPH recreated from the light reactions. Those are going to end up giving electrons now and hydrogens to these particular molecules right here. And now you're going to end up creating G3P. All right. G3P then can go on into the cycle of sugar production. So we go from carbon and the ribulose biphosphate to phosphoglycerate, biphosphoglycerate, glycerol phenaldehyde, which is G3P, and then that goes down into two G3Ps, which ends up becoming sugars. All right. So the sugars in our body, the galactose, the mannose, and all that type of stuff that we get from plants are created by way of this cycle. Now, this part is called carbon fixation. The second part of the cycle is called reduction and sugar production because the products are getting reduced and sugar is getting made. The third part is called regeneration, because they got to make more of this ribulose biphosphate so that they can keep making more and more and more. And all of this happens inside the chloroplast. So photosystem 1 and 2 are in the thylakoid. The dark reactions occur within inside the stroma of the fluid medium that's inside the chloroplast. Let's keep going. Electricity, people, electric foods. This is happening every second that passes by. So let's kind of go through some of what we talked about. All right, we're gonna go through some of the, yeah, just let it play. Yeah, just let it play. We're gonna go through this animation. All right, I like using animations. It just helps us get it a lot better. So we're gonna go through some of this process. All right, so we're talking about photosynthesis. Produces ATP from ADP and PI by using the energy of light to excite electrons that are passed along an electron transport chain, as we talked about before. Coupled with the transfer of electrons is the pumping of hydrogen ions and the splitting of water molecules. We're splitting water and we're splitting hydrogen, people. Nuclear reactors. That's what we're doing every day when we walk around. The plants are doing it too. Introducing photosynthesis. Okay, these are the different complexes that I talked about before. They're probably going to leave out some of the more specific things that I talked about, but it's going to be more generalized so you can just kind of get a visual idea of what I meant. Remember, this is in the membrane of the thylakoid, the emerald tablets, plastoquinone, QB, plasticyanin. Those are the acceptors that I was telling you about. So very shortly, the animation will start, so you'll see the process. Okay, so we got photons, we got chlorophyll molecules, we got protons. Okay, that's the hydrogen without the electron. We got water, H2O, and molecular oxygen, which is 2 atoms of oxygen. NADP plus and NADPH. Okay, remember? And then we got NADP and then PI. They come together, they make ATP. All right, so we're going inside of the chloroplast. Remember, we talked about the thylakoids, right? So as we go into the thylakoids, we're going to go to the thylakoid membrane, where you're going to see all the proteins studded in the membrane. All right, so here we go. This is the complex right here. We're going to start with photosystem 2. Here is a photon coming into the photosystem 2. As it comes into photosystem 2, it's going to start exciting all of these chlorophylls, and what they're doing is they're resonating and they're vibrating like in the Kabbalion, the steps. And what it's going to do is hit the reaction center. When the reaction center gets stimulated, the electron gets passed on to pheophytin and then down to the quinones. Okay, plastoquinone A and B. Okay, once it gets to plastoquinone B, okay, it's waiting on another electron to come down because it's going to pass 2. Okay, there's quinone A, it just received it. Quinone B just received the other one. Those are the 2 protons, those are electrons. Okay, water is splitting. This is showing the water splitting. See, as it splits, the electron gets released. This is how you replenish the reaction center. See, an electron is released every time water is split. So not only are we getting oxygen, but when it's split, it's providing energy or replenishment to the plant as well. Okay, constantly feeding itself. It's amazing. So as we move on, the plastoquinone is carrying the electrons on, and it's going to pass it on to this new complex called cytochrome B6F. As it passes it on to cytochrome B6F, it goes inside the lumen space, and as it goes inside the lumen space, see, these are the electrons. See, they just attracted protons. The protons are going to go up there in the lumen space, and they're going to increase. This is going to create the electrical gradient. All right. Now, this right here is plasticyanin. Plasticyanin takes it to photosystem 1. Here we go with the chlorophylls getting re-energized. This reaction center is now going to secrete another electron. All right. This is ferredoxin. Ferredoxin takes it to FNR, ferredoxin NADP reductase, which then creates NADPH from NADP, which this takes on to the dark reactions that we just talked previously. Now, as this whole process was occurring, as this electron was moving through, all these hydrogens was getting pumped up in this space. They can't get through. The only way they could get through is through ATP synthase. And as it finds this channel, it moves this motor. This motor takes ADP and ATP an inorganic phosphate and turns it into ATP. All right, so let's get back moving again. All right, so photons coming down, protons going out, electrons moving over to photosystem 1. As I said, as it moves over to photosystem 1, then you're going to end up creating the proton gradient. As I said before, protons are going to increase, and we're going to end up showing you the ATP synthase again. All right, this is the ATP synthase. This is a motor inside the cell. It's also in your mitochondria. All right, so here we go. Protons are entering, ADP and phosphate come together, ATP is created. All right, so once again, to summarize it, light comes down, photosystem 2, pushes electrons over to photosystem 1. Increased gradient creates an electrical gradient for ATP synthase to make ATP, and NADP becomes NADPH, becomes a carrier molecule to take on to the Calvin cycle. And you see it's becoming reduced by it changing its color. All right. So this all occurs inside of the plant. Electric foods, people. We have to eat the vegetation. We got to stop playing games. All right, pause it real quick. All right, so now I'm about to take off. How I'm looking on time right now? Got time. Okay, so look. Think about the minds that exist in the community, the youth, that can take that information and turn it into technology. Imagine fueling your home off some broccoli. You know what I'm saying? Or some collard greens. Imagine y'all growing a garden in the back with a pond, and it's fueling your home. Why not? You see all the energy that's occurring when you go into the park and you just look, go in a lawn, and I'm not talking about Buckhead Roundup sprayed lawns. No, I'm talking about East Atlanta, East Point, you know, Brownsville, Harlem lawns, where you got dandelion and plantain growing wild with clover. You know what I'm saying? Growing everywhere. Imagine with just within a square foot of that space, how much energy is made in a year. And what if that energy was stored and then utilized? That's why I want y'all to understand this information. But forget about the future and that. How about just the fact of you consuming it, which we're about to get into now. So let's push on. All right, since we got that out of the way, other unique structures of the cell. Remember, we were talking about the cell. When we go inside the lungs, inside the bronchial tubes, lined inside the bronchial tubes are cilia. These cilia whip back and forth to propel the mucus lining so that as you inhale particulates out the air, they don't go inside your alveoli and you get asthma or any type of process where you can't breathe. So it propels it back upwards. Also, flagellum is another type. Where it's on this bacteria and it helps it to propel. And it's also on sperm. I want you to pause when the sperm come up. So I want to tell these people about their sperm real quick. We have it in our sperm cells. Go ahead and pause that. Check this out. Sperm, right? Let's say I was a sperm, okay? Because sperm's whole point is to find the egg. We don't really understand because sperm is the smallest cell in the body, while the egg is the largest cell in the body. I don't think that's by any coincidence. We never really think about the distance that the sperm has to go in order to fertilize the egg, the type of work it has to do. Well, let's just scale sperm up. Let's say I'm a sperm. What would be the distance to the egg in the fallopian tube if I was a sperm and I had to swim somewhere? How about if I jumped off the coast of California and had to swim to Tahiti? I'm not exaggerating. That's just not off the dome. That's like actual numbers. Sperm have to swim from LA to Tahiti in order to fertilize the egg. And if you scale sperm up to the size of a whale, they're swimming 15,000 miles per hour. Now, not just that, but there's always the conversation in the whole, you know, because in our community, we're black man, black woman always arguing. You know what I mean? We always got a problem. And one of the main arguments we have is the fact that, when a woman gets pregnant, all the work they got to do. But see, if you knew your body, brothers, then you could let her know you already did your work. You swam from LA to Tahiti just to get it done. So now it's her go to get her work in. Let's move it on. All right, so eukaryotic cells, plant animal cells with the nucleus and the membrane enclosed organelles. Prokaryotic cells are a little bit different, okay? Because they're unicellular organisms without a nucleus or membrane enclosed organelles. That's what differentiates them. All cells have a cell membrane, cytoplasm, and genetic material. So let's push on. We're going to talk a little bit about the blood now, people, as we go into the next phase. As we go through our arteries and arterioles and our capillaries, we find ourselves inside the bloodstream. Okay, the bloodstream or our blood vessels are 60,000 miles long, meaning we can go around the planet 2.5 times, all right, with our blood vessels. Not just that, we make 2 million blood cells every second. That is another fact that you need to know, and it cycles our body every single minute. Blood lasts about 120 days, which is 4 months or a third of a year. And they lack a nucleus. Why I have reason to believe that they are the true stem cell. So the blood, hemoglobin, hemogroups, iron, and oxygen. All right. I'm not sure if everybody's able to see the blood very clearly and not on the screen, but we have red blood cells flowing. And what we're going to do is we're going to go into red blood cells so I can explain the properties of it. All right. Get a better idea why we need to get iron. Hemoglobin is the protein inside of our red blood cell, 350 million in each red blood cell. We have 30 trillion red blood cells. Inside of the hemoglobin are hemigroups, 4 per molecule. This is a hemigroup right here. Okay, what is so special about the hemigroup? Hemigroups have iron at a center in order to bind that same oxygen that we got from the plant. See the relationship between blood and chlorophyll? Not just that. Pause that, bring that back a little bit. Just slightly. A little bit back, right there. Okay, so pause it. Hold on. So what I didn't put up here was the molecular structure of chlorophyll is almost an exact replica of the molecular structure of hemoglobin. The difference between the two is the core. At the core of the hemoglobin is iron. At the core of chlorophyll is magnesium. Okay? Now, our body is able to transmutate metals. If you look up Louis Kervran, the transmutation, he talks about that years ago, how our body is able to transmute one element to another element. So if you're eating large amounts of chlorophyll, because remember, I talked about the reflection of light. If something is green, it reflects green, it absorbs red. Well, if something is red, it's the exact opposite. That's how you know they're connected. When you look at the Moorish flag, you see the red and the green. If you just look at color theory, you see they are connected. I'm talking about art class in college. You'll see that. Chlorophyll and hemoglobin have a direct connection. Hemoglobin is what is responsible for giving blood cells its red color. All right. So we could keep going now. All right. So once again, 4 per molecule. Iron is at its core. It is there to bind the oxygen molecule that we saw previously coming out the plant breakdown that I just gave. Oxygen is used in a molecular form in a pair of 2 oxygen atoms. These are the iron atoms in the hemigroup. All right. That's them that bind the oxygen. Okay. Iron attracts oxygen. Blood is our magnetoreceptor. So let's go into the digestive system, y'all. Digestive system is the alimentary canal. It is 30 feet long. It is the height of a tall tree. We're going to start with the esophagus, even though that's not the beginning of digestion. But as you swallow, you have the epiglottis. The epiglottis is a flap that covers the top of your trachea so that you don't end up getting food down your bronchial tubes where you breathe. Okay, the epiglottis moves food down through a process called peristalsis. It's a wave-like motion. I say it's similar to when we were little eating ices and we would get to that last little piece. You can pause it real quick. We get to that last little piece, and when we get to that last little piece, we take our finger and we press our finger one over another so that we get the icy out and we get that last little bit. Well, that's peristalsis, except it's in the opposite direction. Now, as we go through the esophagus, we're going to go through a doorway right here that's called the cardiac sphincter. Well, when you enter through the cardiac sphincter, you enter the stomach, and the stomach has gastric juices, okay? These gastric juices are very, very acidic along with the hydrochloric acid. Now, if our cardiac sphincter relaxes, or we're eating foods that are not compatible with each other, and we get a backup, all right, into the esophagus lining is not structured for the acid as the stomach is. So what happens when acid ends up backing up and touching an area that's not protected, it's going to burn it. And it's called cardiac sphincter. Cardiac means heart. So now you understand why they call it heartburn. Heartburn. Let's keep going. Now the stomach is amazing. It has all these wrinkles because it's able to stretch and grow. You could let it go. You hit play. It can stretch, it can grow, it can keep getting larger, as you see when you're dumping food down there. It has gastric pits. It's going quicker than I thought. Let's go back a little bit. My bad. Back to the stomach real quick. And then we're gonna flow. All right. So as I was saying, the stomach has these gastric pits. These gastric pits end up creating pepsin. Pepsin is where the company Pepsi gets their name from. The reason why they get their name Pepsi from pepsin is because when they first came out, they were using the lining of cow stomach and the hormone pepsin as an ingredient in their cola, along with cola nuts. All right, so that's where the word Pepsi comes from. You ever wonder what their symbol is? We always thought it was yin yang. No, it's not yin yang. The red and the blue represents alkaline and acid, because that's what happens when the fluid is moving around in the stomach. It's the acidity of the hydrochloric acid and the alkalinity of the food and the other hormones that are mixing together. And if you look at the negative space in between the red and blue design, you'll see the shape of a stomach, because that's what pepsin represents. Coca-Cola, on the other hand, dealt with cocaine. And as I spoke about on the other video that I got up with red and blue right now, there's a company called the Stepan Company, S-T-P-A-N, look them up. They're the only company allowed in the country to deal with cocaine. Cocaine leaves are imported into our country legally to this company. They extract cocaine and they put it in Coca-Cola. But they say that it's a non-active form of cocaine. So what they're using it for, right? So then the active part that they say is left over, they sell to a pharmaceutical company. What are pharmaceutical companies doing with cocaine? Look it up. Look it up. Check it out. See it from front. All right, let's keep going. So the next you get into the small intestine. Small intestine is where the action takes place in digestion. More specifically, the duodenum is broken up into the duodenum, the ileum, and the jejunum. All right, the duodenum is where the breakdown and the extraction of the particular elements are taking place, and there's further absorption taking place into the jejunum and the ileum. The ileum leads into the large intestines. We also have the pancreas. We all know the pancreas. The pancreas is where insulin is created. People who have diabetes have a problem creating insulin. All right, it's an exocrine gland and endocrine gland because it deals with both parts of the body. Endocrine dealing with the blood, exocrine dealing with the digestive system. The liver has over 500 functions, but one of its most basic functions is taking poisonous, toxic chemicals and reassembling it into products that we can use. It also formulates bile, which is an emulsification agent. All right, it also stores sugar in the form of glycogen. All right, this is what the liver does. The liver is very, very powerful. We also have the gallbladder, which stores the bile that the liver makes, concentrates it. If it gets overly concentrated through the acidity in the diet, you get gallstones. All right, once again, bile is used to emulsify fats, and we're going to talk about what the emulsification of fats is all about. All right, now we get into the large intestines, and the large intestine starts here at the cecum. All right, the cecum. We also have the appendix, which people say we don't use, but it's actually there to spray any microbes and bacteria that come from the small intestine into the large intestine that should be there. All right. So yeah, of course we got the rectum and in the anus. This is the process in which the food goes through our body. Food never goes in our body because this is a tube. It is outside our body the same way our skin is. We're a donut. And what we want to do is get macromolecules, proteins, fats, and sugars, minerals in our body. And we're going to do that by way of the small intestines. All right. So the small intestines is what we're going to get into shortly. All right. So once again, digestive system, esophagus, stomach, gallbladder, liver, small intestines, large intestines, pancreas. All right. This is the food coming in. All right. As the food come in, you see these little finger-like projections. They're called microvilli. This is what lines your small intestines to increase the surface area. We are flying through the small intestines right now. All right. The small intestines and the microbiome. All right. Let's pause that real quick. When I say small intestines and microbiome, what am I talking about? I'm talking about the fact that your small intestines house I'll put it this way. Let me bring this back. The ratio of bacteria to cells in our body are 10 to 1. All right? Just in our gut alone, all right, E. coli, there's as many E. coli bacteria in our gut than there are people on the planet Earth. There's 8 billion people here. That's how many E. coli. That's just E. coli. But you got home to over 1,000 varieties of bacteria. What are they there for? Well, when we are digesting the plant material, see, this is the misconception that goes on. People say we can't digest plants. No, we can't digest certain parts of the plant. We cannot. That's why cows have 4 stomachs, ruminants. They have to eat, plant, throw up, eat, throw up again. We don't have to do that because we got certain bacteria that consume it for us, and as they consume it, guess what? They synthesize certain chemicals, certain reactions, and a lot of our health in our body has to do with the health of the microbiome because the gut is directly connected to the brain because it's called the enteric nervous system, and you have enteroendocrine cells as well. All right, we can keep going. So as we flying through our small intestines, as I said, they are folds to increase the surface area. So you have the surface area of a tennis court to digest food. Yes, a tennis court. These are all fingers. And even on all these little fingers you see, there are individual fingers on them. Let's pause there. So antibiotics, Western diet, elimination of enteropathogens, vaccinations, cesarean sections, baby formula. What do all these things have in common? These are assaults on your microbiome inside your digestive system. It's one thing to eat good food because most people, they use the excuse that still eat meat. They say, oh man, I done met vegetarian, I got a vegan friend and they sick, man, because they sick. You know what I'm saying? I'm not doing it. Well, how many meat eaters is sick? Huh? How many people eating all that garbage is up in the hospital? That ain't stopping you from eating it? But you be 1 or 2 vegetarians that ain't living up to what you want them to live up to. So then you say, oh man, let me revert back to American Deli. Come on, man. Really? So the problem is that these particular things right here are destroying the microbiome in your body. All right? Let's start here with the baby formula and the cesarean sections. First of all, let's cover EAN so you can see what we're talking about. That's Caesar. That's a Caesar section. All right? Now, what is so detrimental about cesarean section? Well, when a baby is gestating inside the mother, how are they able to get the microbiome? How are they able to get the bacteria that they need? Because they're in the mother and they're cut off. Where do they get it from? They get it from the mother. The mother passes it down to them. But they get it from the mother when they are crowned king. When is a child crowned king? When a mother gives birth to them. When a woman opens up those legs, when she splits those legs and she makes that vesica piscis, and that head starts to poke through. And what do you call that? When a child starts to come through? The stargate. They call it crowning. They call it crowning because you are now done king as you hit this land. And when you come through that vagina, that vaginal canal, you have to go through all these bacteria start interacting with you. And then you also get it when you start feeding off the mother's milk. And when you feed off the mother's milk, you start to activate the digestive juices in your body, your digestive system. And then from there, you get the bacteria and the microbiome. But see, when you get a cesarean section, they slice on the side and yank the baby out. The baby's shook and traumatized for life and don't get the bacteria. So where do they get their first bacteria from? They get it from the dirty doctor. You know what I'm saying? I don't care about them gloves and that mask, man. That ain't doing nothing. Baby formula gives them their bacteria. So now you have bacteria that's made in a lab specifically designed to attack the beneficial gut flora in your body. So that's why when a child starts getting 2, 3, 4 ear infections, then they start giving them what? Vaccinations. You see what I'm saying? Then they give you a diet that eliminates enteropathogens, right? Okay. The Western diet, meaning the diet that's over here in America, antibiotics, all these mechanisms right here destroy the microbiome. All right, let's push on. Okay, so like I said, we flowing through. Got to get up in here, microvilla. All right. So the microvilla inside the duodenum. As I said, there's fingers on fingers. All right. This is where the absorption takes place as the mechanisms start to break down. All right. This is the intestinal lumen or the tubular space. All right. Inside. Now, this is the gut mucosa. This is actually the ileum of the small intestine. All right. The Peyer's patches, okay, or aggregated lymphoid nodules, or occasionally PP for brevity, or organized lymphoid nodules, aggregations of lymphoid tissues that are usually found in the lowest portion of the small intestine, the ileum in humans. As such, they differentiate. Yeah, you already was on it with me. Appreciate it. They differentiate the ileum from the duodenum and the jejunum. The duodenum can be identified by Brunner's glands. The jejunum has neither Brunner's glands nor Peyer's patches. Because the lumen of the gastrointestinal tract is exposed to external environment, like I said, it's a tube. It's not inside, it's outside. Much of it's populated with potentially pathogenic microorganisms. Peyer's patches thus establish their importance in the immune surveillance of the intestinal lumen and in facilitating the generation of the immune response within the mucosa. So we are looking at the ileum, which is the third section of the small intestines. We're going to go back into the duodenum when we talk about the liver, the pancreas, and the gallbladder. But right now we're talking about the ileum, where these Peyer's patches are. These are different nodules that are situated within the villus, inside the ileum, that is responsible for dealing with all of the harmful bacteria and harmful particulates that made it through the food that you just ate or just came inside your body because it's open to the elements. All right, let's keep it going. Keep it going. Keep it going. All right. Pushing on, pushing on. Yeah, man, we got to see our body, man. We call ourselves God body. We need to know why we got body. Why do we use that term? What does that mean? You can't call yourself that if you don't know yourself. You know what I mean? So let's go back down inside. You know what I mean? Chewing your food, you swallow your food down the esophagus, through the stomach, into the small intestines, as I stated before. It's like Greenleaf Herbal Solutions is our company. My mother is the founder, studying under Sapien. All right. So as we cruise through the ileum, we see that it's studied with the finger-like projections called the villi to increase the surface area to absorb the macromolecules of sugars, fats, and proteins. All right. Now, as we find this clearing, we're going to fly around and find a nice spot to land our helicopter in, submarine, whatever you want to be in at the moment. Okay. And this nodule, as we split it, we're going to see that it's connected to the villus. Okay. So we got the villi right here, and we have the Peyer's patch right here. Okay. And both of these mechanisms end up dumping into the mesocentric lymph node, which sits right here. And this dumps into the lymphatic system. Okay. Now this is a crypt. The crypt is where stem cells are to replenish the villus. You constantly are making new cells. Cells are being created. down there in the crypt. Oh no, you could keep going. It's okay. That's the epithelium, the lining. All right. So let's zoom inside the Peyer's patch. In here, you have B and T cells. All right. This is your immune system that's inside this Peyer patch. That's a macrophage. All right. This is an M cell. M cell sits inside. These are epithelial cells. Okay, with the microvilla sticking outside. This M cell regulates what goes in and out. See, things can come inside the M cell and then go past these cells to get inside. The macrophage consumes it. So if it's not good for being inside, it's going to destroy it, similar to the lysosomes do. This dendritic cell is going to create a foot or a hand and reach out into the particulates that are flowing through this mucus lining and bring them inside and consume them. Now this one actually got hit with that thing. That's why it's starting to break down, and you're starting to see all the chemicals come out of it. All right, but what it does is it attaches to a T cell, and the T cell then is going to be able to transfer the information on to someone else so that it can take care of this particular situation. So you see, it went and it traveled up now to the villi. So now we're actually in the microvilli. Okay, I mean, we're now in the villus, the finger-like projection. So you see, it came up here, and it's all of them are going through the same process right now. And the process is to control the situation so that it doesn't harm the ileum, because remember, the ileum leads into the large intestines, and a lot of us deal with colon cancer. Now look, they got word that this was coming. Okay, so this black represents a bad substance in the body. You know, you probably just went to KFC. You just got one of them antibiotics that GMO just infiltrated. See, as it's starting to destroy the cells, they're separating, okay? And it's leaking, just like leaky gut. So now all these little particulates are emptying inside, and it's starting to infect the villi. Everything is popping off now. So what is it going to do? How is it going to handle this situation? Because it's worsening. Do you see? It's starting to propagate throughout here. It's getting darker and darker. All right? Everything is getting infected. But see, your body is prepared for everything. Your body is going to figure out a way to take care of this situation. Okay? Tumor necrosis factor, interferon gamma, those particular properties are there to create a circumstance to deal with this matter. So it brings a neutrophil out. The neutrophil, he's the gangster. He's the super henchman. Look what the neutrophil gonna do. The neutrophil like, all right, I see what you're doing. You ain't ready for this. Ugh. So he did the suicide bomber. No offense. But he straight strapped the bomb on and Allahu Akbar and blew up and tore out the whole problem. So that thing that you got from that KFC got destroyed. The cells are now able to regenerate, and now all the other cells are now able to heal themselves because they're good. Now these macrophages are going to consume all the cells that have broken down and died. Any of the cells that destroyed, they get replaced. Those crypts that you saw at the bottom create new cells. That's why you just saw it slide over like that. So this is happening right now in your ileum. Every moment, every day. That's why you got to keep giving your body energy and electricity so it keeps working. There's so many things happening in the body all the time. You can't keep track of it. But what you can do is make sure that you're looking out. All right, now you keep going. We good. So microvilla, you know, when I'm talking about the products we sell at Greenleaf Herbal Solutions, you deal with properties that go inside of your lumen, and they're able to clean it out. You know, that's what you see here. As you see all these little white balls getting absorbed and pulled into. Okay, they're pulling all of these toxins and these poisons from around. Okay. All right, so bile and emulsification. What is bile and what is emulsification? Bile is formulated inside the liver. It is sent up to the gallbladder to get concentrated so that it can be used to emulsify fats, because when you eat fried chicken or any type of oil, when it comes inside, it's an aqueous solution in your intestines. It's water. Water and oil don't mix. So how do you break down oil and water? Well, you need an emulsifier. What is an emulsifier? Well, if you got a greasy pan, you get some Ajax, and you get that oil and that grit out, because it's able to deal with both water and oil. Okay, it's hydrophilic and hydrophobic, meaning it's amphiphilic or amphipathic, meaning it likes water and oil. And what that does is it breaks the oil up. So bile breaks the oil up, or the oil globules, into smaller globulets, emulsification droplets, which then can be dealt with by way of micelles, which are these little mechanisms that can then capture the broken down lipids, take them to the walls of your intestines so that your intestines can then absorb the fatty acids and the glycerol, which are the micromolecular states of fat. Now, these green balls represent phytosterols. These are the healthy cholesterol that you get from plants. And when you eat them, then you form micelles that are more beneficial to the body. Okay, so along with bile salts and these plant sterols, the fats are able to get transported in the lumen to the wall where the villi are, so that then they can be absorbed and get inside the bloodstream or the lymphatic system and go wherever it needs to go. All right, so we're going to see another example of that in this other animation, because I want you to understand what it means. And remember, cholesterol, when it's not balanced and you're not taking care of it right, it's going to affect the bloodstream. It's going to affect the bloodstream in such a way that it can create arteriosclerosis. You know, arteriosclerosis is the calcification and the buildup plaque of the arteries, which you don't want, and it also will affect the heart. Now, cholesterol isn't bad, you need it, but it's best to get cholesterol from plant-based. You don't want to get it from animal fat. Okay, that's how you create LDL, which is low-density lipoproteins, which creates a real problem. So once again, here you go. Cholesterol is entering in the duodenum. Okay, as it enters inside the duodenum, you know, you want to form something called a micelle. But see, micelles are water-soluble packets which transport cholesterol to the intestinal wall. As you see, it comes to the intestinal wall, you know, breaks up, and it's going to go through the intestinal wall, and it's going to go inside the blood. But as I said before, if you're dealing with animal cholesterol, you'll create arteriosclerosis, which is this plaque, which will narrow the pathway of the blood to flow, which will slow it down, which would increase your blood pressure. Okay, that's how you get high blood pressure, because the pathway 285, there's an accident blocking 3 lanes, and that backs up all the way back to Glenwood, so you can't get home in time to see your wife or your kids' rehearsal or whatever. But when you have plant sterols or plant-based sterols, which you get from our products or you get from a plant-based diet that you're eating, then you create a more effective form of a micelle, which then, when it gets inside of your blood, now the type of cholesterol that's getting transported, the chylomicrons, are in the form of high-density lipoproteins. High-density lipoproteins are way more better in regards to flowing through the bloodstream. They're not falling and against the walls and building up. Okay? So that's what you want to deal with. All right? So let's get into the liver. How we looking? We good? Okay. All right, so let's go into the liver. The liver is powerful. I can't even talk about all the jobs that it do. Like I said, it's like 500, maybe even 5,000 different things the liver does. It takes up most of the portion of your body. It's like the upper right side of your torso, and it takes up about 1/3 of the space. It's crazy. All right. Now, we have the portal vein, all right, which leads to the spleen, the pancreas, and the small intestine. A lot of people don't know what the spleen do. Let me pause that real quick. The spleen is responsible for the health of the blood. It's a storage area for blood. So if you hemorrhage or you're having a blood issue, it'll get reserves from the spleen. But most importantly, blood goes through a 120-day life cycle, meaning it lasts for 4 months. And this is only when you're healthy. I mean, it's going to last a lot shorter if you're not doing the right thing. But if you're on it, then it's going to last for 4 months. And when it lasts for 4 months, it's going to go see the spleen. And the spleen is sitting there like Don Corleone, and he's looking at him, and he's like, yo, you think you fit to keep this thing going? And the blood cell is like, yeah, I got it. And he's like, we'll see. And the spleen is made up of these different type of filters. See, blood is kind of like mice. You ever notice that mice or rats could be huge, but they can get into the smallest holes because they can dislocate their bones and fit through it and then put them back together and go where they need to go. Red blood cells do the same thing. They're able to change their shape and bend it in certain areas so they can get in real narrow spaces. Well, the spaces in the spleen are so narrow and so fine-tuned that if that red blood cell is not fit and don't got it, it's like the combine before the draft, right? They gotta run the meters, they gotta pass all them tests in order for them to get drafted. Well, the red blood cells need to get drafted. All right, but guess what? If they don't make them numbers, then they're going to explode. They're going to bust. And the contents of the red blood cell that got destroyed gets recycled by way of the liver. If it passes through, the red blood cell continues to live. So that's what the spleen does. So we can keep going, brother. All right. So we got the hepatic artery, all right, and the celiac trunk, okay? So we're going to start getting into the different energies. So yeah, oxygen-rich, nutrient-poor with the artery, but with the veins, it's oxygen-poor but nutrient-rich. The veins don't have pumps around them like arteries. They have valves, so the blood moves through your veins through your motion. So if you're not running or jogging or in any active condition, that's how you get varicose veins and stagnant blood, poor blood, because that's how your veins move the blood around, okay? The gallbladder, as I said, exocrine gland dealing with the digestive system. The bile comes in there, it gets stored, it concentrates it. Here it is secreting the bile as the oil comes down into the duodenum so that it can emulsify those fats. All right? And after that happens, it comes through the portal vein into the liver. The liver is made up of lobules. The lobules are these hexagons. They're shaped just like this. You have the artery, the vein, and the bile duct. We have the central vein in the middle. Okay. Got to know about your liver because we got a lot of liver diseases out there because we love to drink as people. We be knocking that yak back, drinking that beer every day. Portal vein, hepatic artery, and bile duct is what it is, and it's called the portal triad. All right. As you split it, you see it's a hexagon, and they're made up of these different hexagons. All right. And it's funny because it's almost sacred geometry involved in all of this. Okay. Because you're going to see how they relate to one another. All right. So the classic lobule is represented by these 6 artery arrangement. All right. As you see. Kupffer cells, pause that real quick. Kupffer cells are these little cells right here. There's particular macrophages that sit in this area as the blood comes through. And what it does is it consumes some of the blood that isn't up to par. It kind of works hand in hand with the spleen, where it consumes it and it uses products in order to help to create the bile salts. So this is how your blood ends up helping in so many different ways. Let's go. Let's keep it going. All right. Now this is called, okay, as we said, it's a classic lobule, portal vein in the center, artery, vein, and bile duct. And there's 6 of them around it. And then how the flow takes place is as the constituents and the flow comes through the vein, it comes in there. Okay. It comes and it dumps inside. So that's what those arrows are talking about. Now, a liver acinus is the relationship between 2 livers, lobules together, and it makes a diamond formation between one portal vein and another portal vein. All right? Like I said, this is sacred geometry. Vesica Pisces. You know, when I study sacred geometry, I see these shapes all the time, and this occurs in your liver. But see, when you are consuming, oh yeah, and then also Remember, glucose is stored as glycogen in the liver. So in those times where all the sugar's been used up, then your body is able to take the glycogen and break it down back into sugar in your body so that you can feed during your fasting cycle. So you can still have sugar when needed, because you need sugar sometimes when you're not eating. So how are you doing that? You're doing that because the liver is supplying the sugar by means of glycogen. All right. So glucose to glycogen, back to glucose again. And this is done hand in hand with the pancreas through a mechanism, enzyme called, or hormone called glucagon. All right. This is representing the flow importance or strength. Rather, this is the most strongest flow, then that's the second strongest, and then the third. Once again, these are the lobules. These are how they relate to one another. Okay. So many hepatic diseases out here. We got to put that alcohol down. It's really not necessary, y'all. I know everybody want to get their drink on, but I don't know if we're supposed to be popping bottles right now. You know what I mean? So yeah, that's the anatomical relationship. Okay, and this is the functional relationship. Okay, how they relate to one another. Okay, so here we go. As I split this open, see how the blood flows. It's all dumping in there. All right, so now we're going to get into insulin. People, let's get into some diabetes. We're starting to get close to the end. Okay, part one, role of insulin in your body. Insulin 1 and insulin 2. We're going to talk about insulin 1. The difference between insulin 1 and insulin 2 is the location of the issue. Insulin 1 deals with the location being in the pancreas. Diabetes 1 deals with the problem being at the site of the pancreas. Diabetes 2 deal with the site of the cells that the insulin is going. So islets are containing beta cells. These are beta cells. Beta cells are where insulin is produced. This is the islets of Langerhans. Islets of Langerhans has alpha, beta, and delta cells, but the beta cells is what secretes the insulin. Glucagon is secreted by the alpha cells, which help to turn glycogen back into the glucose. So the glucose arrives by way of the capillary. As the glucose comes by way of the blood, remember we digested food, we got glucose in our bloodstream. What it does is going to stimulate the beta cells. And as it stimulates the beta cells are going to start to secrete insulin. This is it. Insulin is starting to pop up, right? And that's the signal. Okay, just like if you're trying to watch the fight, you need a signal coming to your satellite so you can see the game, right? So insulin starts to travel through the bloodstream as you see it cascading down. Where is it going? It's looking for the cells because the glucose has to go in the cells so you can get energy, all right? So as it's coming down, these capillaries are coming down, these arteries deeper and deeper into the body, they're looking for the cells. As we zoom out, we're going to look at the muscle fibers. These are the muscle fibers with the blood. And if you look up here, the blue, that's the insulin coming, right? Propagating, cascading, coming down, all right? As it comes down into the muscle fibers, we're now going to go to the surface of the cellular membrane so we can see the receptor sites, all right? And the receptors that receive the insulin signal. These are the insulin receptors, okay? The insulin receptors are waiting for the insulin to arrive. As the insulin comes down, starts cascading down as a signal, as a radio wave signal, they start to take in the signal. As they take in the signal on the other side of the cellular membrane, they're going to create a cascade, a signaling cascade to the nucleus to tell them to let glucose in. And they do this by creating transport channels. That's what these purple things are. Now all the glucose, these little white dots, now can get inside the cell. And as they get inside the cell, they can get to the mitochondria. And then guess what? You can start getting into cellular respiration, and you can get into the electron transport chain to create ATP and energize the body. So how does diabetes 1 occur? Well, because of a poor diet, what happens is you start to destroy your beta cells. Your beta cells are not getting the nutrients they need, and they're starting to get seen as a problem. And your white blood cells are creating inflammation response chemicals, and it's causing the inflammation to occur, and then for macrophages and other things to start consuming the beta cells and breaking it down until your beta cells shrink and they become maybe a quarter of the size. So now because they're so small, when you eating all the sugar, guess what? How is these little beta cells gonna secrete enough insulin to deal with a stack of pancakes in the morning, a milkshake, ice cream, hamburgers, or spaghetti, which is starch, mashed potatoes, macaroni and cheese? How's it gonna deal with all of this shit? When it done shrunk down that small. So you see the healthy, you see the small. I want y'all to see what goes on with diabetes so you can understand it can be cured. It could be reversed. All right? So now we're going to deal with diabetes 2. Okay? Now, the funny thing here is as the beta cells start to secrete insulin, okay, we're showing the process again. This is a similar animation. Okay? These are white blood cells. Okay? Now, as we always think white blood cells are good for us, they're always there when we have a problem, which I don't think is a good thing. If I have an issue and every time there's a problem, I see a nigga there, something wrong with that. You know what I'm saying? So, what you're going to see shortly is these white blood cells are going to start to secrete an inflammation response through these orange. And that's going to cause a turn of events, which is not good. All right? So we're going to go inside of the white blood cell so we can see what's really going on here. All right? See, the mitochondria is creating free radicals now. Free radicals are not good because they cause havoc in the body because there's no control in them. They can do what they want to do. Now, these free radicals come in. They infect the sentinel proteins. Sentinel proteins then get a response from the free radicals to assemble. What are they going to assemble into? Well, we're going to see what they're going to assemble into because this ain't no joke. This goes on every time you got inflammation. I had some brother that just caught the epiphany on how detrimental inflammation is to the body. So they form into the structure. And as they form into the structure, they get adapter proteins that come onto it. These adapter proteins then complete the structure. And what they do is they're going to start to change proteins that exist inside by activating them. You see, as they turn into orange, these are different proteins activated to create the inflammation response in your body. And wherever there's inflammation, you're going to find a gang of gangbusters up in there, which is going to create pus and problems and disease, which could turn into an issue. And disease only deals with the site that inflammation is occurring. All right? So here we go. Activation of inflammatory protein. This is going on every time you knocking out that hot dog, which ain't nothing but a combination of cow eyelids and pig dick. So all the leftover parts, the teeth, the cheek, the ass, like, leave it alone. So the inflammatory proteins are starting to get secreted. And as they start getting secreted, they're going to start shrinking the beta cells. Okay, beta cell death. And if the beta cells start getting destroyed, then the beta cells are going to shrink. Okay, and I'm still on diabetes 1 actually right now. Okay, so you see the islets are going to start to shrink, going to start to get smaller. Okay, if the beta cell becomes too small, if you're trying to scream to somebody and get their attention, you lose your voice, how are you going to get their attention? Right? That's what's going on here. And this all occurred because of these white blood cells, man. These white blood cells did it. Okay, so an insufficient amount of insulin is being produced as the glucose is coming down through the bloodstream. So as it goes to the cell sites, guess what? So this is the cell surface magnified 7 million times. So the insulin binds, right? It's binding. So this is the problem with diabetes 2. Diabetes 2 is a problem at the receptor site. It means you're producing enough insulin, but the receptor is not working properly. And because it's not working properly, then it can't tell the nucleus to open up and let the sugar in. And if you can't let the sugar into the cell, the sugar stays in the blood. The liver could store but so much in the form of glycogen, but so much in the form of fat. And then from that point, it goes in other areas of the body and destroy. Okay? This is inside of the cell, the other side of cellular membrane. These are insulin receptors that start the signaling cascade. When it works properly, there's receptor activation. See, they come together, they link, and when they link, they create a signaling cascade of other proteins that go from the cell membrane to the nucleus, and then the nucleus back. You can tell they're working properly because they're green, they're glowing, they dancing, they moving. All right? But what we're going to see shortly is that when there's a problem at the receptor site, it's going to be in black and white. Okay, so we're seeing here protein interaction and activation. Okay, signaling cascade. See, they're coming together and they're going to start to create a cascade, one protein to another. I mean, you're not going to tell me that this happened by chance, that there's no intelligent design. I'm sorry to tell y'all that, people. There's too much science here for me to think that this just happened by chance. There's something involved. Creation needs a creator. So normally, this is what would happen. Type 2 diabetes, this is what would happen. It's gray. Now, this is information that Sadie been talking about, my mama been talking about. But see, now science and animation has caught up to show it to us. So right here, you see that they cannot connect. They cannot activate. If they can't pause that, because they can't activate, they cannot create the signaling cascade. Bring that back a little bit. All right. So to sum it up, diabetes 1 deals with the islet Langerhans beta cells shrinking, being consumed, not producing enough insulin to create a sufficient response for your cells to let sugar in, okay? Diabetes 2 deals with enough insulin being produced, signal coming in, but guess what? It's not being received. When I say receptor sites on the cell, I want you to think of a cell phone, okay? Because our cells are cell phones. How do our cell phones work? I dial a number, I call somebody up, okay? Relaying a message. Well, guess what? Say I go in an area that has no signal. I could dial the number all day. My phone works. I work. But guess what? The signal's not getting through. If the signal doesn't get through, then the person can't receive my call. And that's what's going on here. It's just that simple. So what do? You got to clean the body out, get the acidity out, because acidity is buffered by calcium ions. Calcium ions is what creates alkalinity in the body. But if you're constantly acidic, all right, because you got an influx of nitrogen and urea and uric acid, because that's what flesh is going to introduce into your body. All that gravy that you like ain't nothing but pus and blood and urea. Simply put, that's all it is. I'm not saying it don't taste good, but that's what it is. So when you get all that in your body and the acidity level goes up, then you have these little organ cells within inside your bones called osteoclasts that break the bone down. And as they break the bone down, it releases calcium ions to buffer the acidity to raise the alkalinity level up. But if you keep bringing acidity in your body, then your osteoclast is going to get overworked and you're going to have deteriorating bone. You're going to break your bone all the way down. You also have osteoblasts that build the bone back up. But guess what? Your calcium ion levels are dropping down. So now when it tries to replaster your bone, when it tries to regenerate new bone, the bone is all spongy. It's too holy. It's not as strong as it used to be. Just like the wood that's sold in Home Depot today ain't the same wood of the 70s, because they're taking the young saplings in the trees because they're trying to get to answer the demand. So now when we build homes, the homes don't have the same infrastructure and strength as they did back in the day because the wood is not given enough time to mature. All right, let's go. So as I talked about earlier with the enteroendocrine cells, let me pause real quick to give an introduction of what we're about to do here. Now, we'll pause that real quick. Appreciate you. So the enteroendocrine cells are specialized cells which inside the small intestines, okay, or inside the digestive system. And what they do is they're responsible for secreting particular hormones that stimulate the secretion of cholecystokinin, which is what stimulates the secretion of bile, secretin, trypsin, all those particular hormones and chemicals that need to get secreted during digestion are done so by way of the enteroendocrine cells. They are directly connected to the nervous system. This has always been speculated, but until now, we actually have the actual picture of it through a new process where I think it's called block spectroscopy, something like that, where they end up taking different two-dimensional pictures, putting it together, creating a three-dimensional block, and then extracting the negative space to see the three-dimensional structures in it. And in there, they were able to finally get a full composite of an enteroendocrine cell. And when they did that, they ended up finding out that it had an axon-like appendage on it. So to find a cell that secretes hormones, to have an axon like a nerve on it, is unlike anything else in the body. And then to see it connected to the glial that deals with the nervous system, which shows that if you eat food that are GMO, or you eat food that has viruses or bacteria in it that is not compatible with your body, then it's not only going to tear up the digestive system, okay, but because of this enteroendocrine cell and the fact it has this neuropod, it can get directly connected to your brain by way of the nervous system. So if you get that in your stomach, it goes to your brain as fast as a signal does. And that same virus that was in your gut is now in your brain, driving you crazy, and you might not even know it. So this is the new information that they're coming across. So you really got to be mindful of what you eat. This also proves that your microbiome, your intestines, is your brain. As is above, so below. We learned about the law of correspondence. We're talking about the Emerald Tablet and the Kabbalion. The law of correspondence is the second law after the law of mentalism. As is above, so below. So if there's a brain in my head, there must be a brain down below. And the brain down below is our intestines. And you know this because there are neurons that are embedded inside of our intestines that directly connect to our brain. That's why a way to a man's heart is through his stomach. You hear what I'm saying? And the same thing with a woman. So we got to be mindful of what we eat. That's why I spent all that time talking about the plants in the beginning, so you can understand that when you take in that vegetation, that electricity, you don't got to worry about the rest of this trash and this garbage. Just get it in, you know? All right, let's go. All right, so we're going to see the process. Microtome, it went too quick. Okay, block surface, that's what it's called. So they take a picture of the scan, all right, and it's just two-dimensional, but they take multi-pictures of different slices of this area. They put it together, they make the block, and then they extract it out, okay? And when they extract the negative space out, you see all of these cells that are inside the intestines, but inside there is one enteroendocrine cell. So we remove the rest of the cells, we see them, and he's sitting right there. Now, these little things right here represents villi, microvilli that enters into the lumen. This is in the base, okay? This is the axon, all right? Axons are only on neurons, though. What is the axon doing on a cell in the intestines? Okay, that's the nucleus. These are the mitochondria. These are the secretory vesicles. Look at all the vesicles that secrete hormones. All right, these are the filaments. All right, these are the same filaments that are inside axons that transport neurochemicals. All right, like I said, this is what points into the lumen, the microvilli, and the intestines. This is embedded inside with the other. All right, this is the neuroglia. All right, this is what connects to the nervous system, enteroglia. Okay. Once again, this is the axon right here. This is what connects to the enteroglia, which directly connects to our brain. So we have to be mindful of what we eat and when we think. So let's talk about some neurons, okay? We about to wrap it up, y'all. We almost there. We good? All right. So we got to understand that it's all about mind. First law of the Kabbalion is mentalism. All is mind. What do you mean by that? Whatever you want to do, you only got to think it. And then you back it up with the action. Because when we go inside the brain, it's a wide space. And neurons are capable of growing and developing new connections all the time. This is called a dendrite, growing out of a neuron, which represents tree. And that's where we receive information. Soma is the body where it's processed. Axon is where, you're good. Axon is where the information is propagated and continues on to the following axon. All right? You only got one axon, but you got many dendrites. What we don't know is that as we develop memory and we learn new things, we develop more connections and our axons continue to grow. They grow to find other areas to connect to so that the thought, the idea manifests. And as you look in the brain and you see all these neurons everywhere, how does this neuron know where to go? And it's interesting because it's shaped like a hand and our neurons are reaching. They're looking for a destination, all right? And you have all these different connections happening all the time. That's why it's good to get out in the environment, breathe, go to the park and meditate and go and travel and learn new things because you're going to create new connections. And how do I know how to make a new connection? How does my neuron know where to go? Because you have guidance proteins. Just like you got guidance in your life and guiding light that helps you to find the path of least resistance where you need to go. And you bob and you weave and you find all types of things in your way, but eventually you find the connection that you're looking for. And when the neuron finds the connection he's looking for, the axon, it'll form into the synaptic knob and it will connect. And as it connects to the synaptic knob, you make your connection. So whatever you think, you will manifest in reality. Real talk, facts. This goes on every second, every moment of the day, people. And with that, I say, Ashe.
All right, so I want you to give the people a summary real quick.
Sure, yeah, no doubt.
And just turn this off.
All right, so today we went into a lot. I got a little deep. I don't know how deep I got. Most people, you got people out there that go all the way in, and some people was for the first time. But pretty much what I came today was to reiterate what I learned. I came underneath the feet of my father, George Edward Tate, poet laureate of Harlem, who deals with African nationalism. He deals with our people, the red, black, and green, our culture, Africa. All right. He's the first one to put me on the stage to speak in front of the people. I got to speak for John Henry Clark and Dr. Ben when I was 6 years old, reciting the poem, I'm a Black Man. I knew from a very young age who I was. I sat at the feet of my mother, Annette Thomas, who sat at the feet of Dr. Sabi, down in Honduras, down in St. Croix, in Brooklyn, New York, Usha Institute. All right, where we learned about electric foods. We learned about the health. The elders in my life are in their 60s. They're in their 70s. They're in their 80s. Sabi has 2 children. He's 80-something years old. He got like a 1-year-old son and like a 3-year-old daughter playing with him in Honduras. You know what I'm saying? My father, he's in his 70s. He still takes care of his business. I can't get over him to this day. He knows what time it is. My mother, y'all know Mama Pill. Y'all see how she glow and what she look like. And then I got other elders that I build with that y'all haven't even seen that are also in their 80s. So I deal with a lot of people that take care of their body. Okay, they know how to use their mental faculties in order to extract the energies out of the food that they're eating. I wanted to be able to show you visually what electric foods were all about. So you know that when you go out and you make these decisions, okay, yeah, you might eat some cake and you might throw a chicken wing down or whatever. But guess what? The holidays are coming. Okay? They're coming at you first with Halloween, where they're going to sugar you up and your baby up. Okay? That's going to get the super sugar rush. The next thing that's going to happen, they're going to hit you with Thanksgiving. And that's going to just hit you from all types of angles, the overeating. All right, which is going to knock. Remember, the pancreas got worked. So you can't even use the pancreas on Thanksgiving. Now it's all up to the liver. All right, but we drinking. We drinking then, and we're going to drink on New Year's. All right, Christmas is going to come around. We're going to eat every single day for 12 days, right? And then it's going to roll around into the Valentine's Day with the chocolate again. So they're going to start with the sugar, and they're going to end with the sugar. So that's why I'm trying to get y'all to understand, prepare your bodies. If we're in a war, how are you going to win a war when you're out of breath? All right? So this is going to be my closing remark. I'm going to reiterate the same thing that me and my mother have been talking about. Look, I have nothing but love for the elders of the community. I appreciate all the work they've done for them. I would not be here. They are my ancestors. The elders of the community were so amazing in these past years in regards to the information that they gave to get this conscious community started, that in pushing and investing their time, they actually sacrificed their health in the process of doing it. That's why they're dropping like flies. Now, when we look at the elders in our community, one conversation you do not see consistent, and that is that of health. We do not hear that conversation. Well, I'm here to resurrect that conversation of health, okay? Because my elders, they're getting up there, and I'm not afraid of them going anywhere. They might just want to go to the islands and relax and eat papaya all day. You know, it's time for me, the youth, as well as those of you out there who resonate with me, who feeling me, to get up and get this ball rolling so we can help all these people who's willing to fight, strap up, and do their work. They need to have the health behind it for stamina and endurance. That's the only way we're going to win. Because when you look at the amount of energy that's invested by way of the beast in regards to what is taking us out, just look at the food industry. Look at the pharmaceutical industry. And then you're going to see what they're really putting all their money into. And if that's the case, then that means we need to checkmate by being one step ahead of them, by not falling victim to the sell your soul food. Peace.
Peace. Thank you guys for tuning in. We want to say peace.
Oh yeah, I gotta. My bad. I'm tripping. I gotta greenleaf-herbs.com. Okay, that's the site of Mama Pill, because everything that I just talked about, if you need the boost, if you need the energy, if you need the products, if you need the love and the light in your body to get yourself out the rut and get yourself going, then you got to get the age-old tradition of the wild-crafted botanical tonics, therapeutics that my mother, these are the same tonics that my mother used when helping to cure the person of AIDS in 1987, which she and Dr. Sabi won the court case over. So because of that court case, by law, we can use the term natural vegetation cell food in order to take care of our community. Just don't use the term pharmaceutical or drug or anything like that. Use natural vegetation cell food. That's how you help your people. Okay, the TomMaryTemple.org, that's my father's site that deals with culture and learning more about your history and tracing our heritage back. We got Ocean 14 with Blue Pill. We got KTL Kings County with Red Pill, KTL Media. You got Hollywood Decoded with me, KT the Arch Degree. You can contact me at kt.kamani@gmail, and I'm Kamani Tate at Facebook. All right, so I think this will be my last sign off.
All right, family. Thank you for watching. This was a powerful, powerful Black News 44 Studios presentation by none other than KT the Arch Degree. We want you guys to hit the subscribe button for more content. We're going to have more KT back out here again with another powerful PowerPoint presentation. If you want them, you got to leave some comments, man. You got to subscribe. Let us know that this is the content information you wanted. We want to give you a nice professional presentation. No interruptions, nobody cutting them off, no distractions, because we want you guys to get this real powerful info. And we hope it was something that was life-changing and will help to raise the frequency of consciousness that you can now share with others now that we got this information. So with that being said, we want to say peace, love, shalom, hotep, assalamu alaikum, Black Power family, all right? We love you. Stay tuned, subscribe, and we'll speak to you again. Peace, y'all.
Peace. All right.